Hello and welcome to a new episode of the Green Innovation Group podcast. We are delighted that today we have a guest in the studio. So welcome to the studio, Luisa. I'm really excited to have you here, so thank you so much for coming. We debated a little bit if I should bring a present for you, and you said I wasn't allowed to because dissemination is actually a part of what you do as a scientist. Scientist, huh?
Scientist in the making. Like I'm still doing my PhD, so I feel bad calling myself a full scientist.
Really? Is that imposter syndrome, or is that like for real?
I think it's a mixture of the two, you know, because I actually Googled trying to figure out if I could call myself a scientist. And some sources said that because I don't have my PhD yet, I can't call myself a scientist or a researcher. Wow. But then I also do think it is a bit of imposter syndrome, because I know many others who call themselves scientists who are still doing their PhD or postdocs or something like that. But I do think postdocs are actually more allowed to call themselves scientists because they do have their PhD degree.
I didn't expect the conversation to go this way, but for me you're a scientist, and then people can hit me in the head with whichever accusations they'll have afterwards. So please feel very free to do that. We're going to talk about the microbiome today, and that's because you're an incredibly smart person and you know a lot about the microbiome. And I first listened to you describing this in Den Dyriske Time, a podcast that our mutual friend Alexander is hosting, and I was just thinking, this is fascinating. I wanna sort of have a deep dive in this, and also, of course, educate myself, but also learn more about how does this relate into a health context. You talked a lot about sort of into an animal setting, animal context. To set the stage, could you please explain to me what is a microbiome, and explain it as if you do it to an 11-year-old, because that's the capacity I can muster right now.
That's your listeners, or?
No, that's me. That's me.
I was just joking. So the microbiome is a community of microbes. The microbes can be bacteria, viruses, nematodes, fungi.
And nematodes, we know those?
They're kind of worms. So it's like, it can be small worms, microscopic worms. But what they all have in common in this microbial community is that they're microscopic small, and they are everywhere. So you can find them inside your gut, you can find them in the soil, you can find them in corals, and so on. They're all around us.
And it's basically, I mean, it's from top to toe. Where does it start and where does it end, the microbiome in a human?
Oh, I think it's difficult to say like where it starts and where it ends, but I mean, we can maybe start with life. So the way we reproduce, our baby goes through a woman's vagina. So that could be the start of your microbiome, because scientists don't believe that you are exposed to many microbes inside what's the word?
As a fetus or
yes, exactly. As a fetus, yeah. So when then the woman gives birth, you're then first exposed to the woman's vagina's microbes. And that's when your microbiome starts to develop.
And that's why they do the thing when you have the, what's it called? When you don't give vaginal birth, but you...
The C-section.
The C-section, yeah. And you cover sort of with some of the...
the mucus from the vagina.
The mucus from the vagina in order to protect the baby and develop the immune system.
Exactly. Some do that. I don't think yet it's anything doctors actively do in all hospitals. It's not like the usual procedure when having had a C-section. But I do think we in the future might see that it will become the go-to. Like that's what you do when having a C-section to prevent developing autoimmune diseases.
And basically, so that's sort of the start of the microbiome, and then it develops for the first couple of years when the baby is in touch with different bacteria and virus, and they take it in, and somehow that culture is built up during the first how many years of a human life?
So it's still debated, but many believe that it's from the age of 1 to 3 that you really recruit your microbes from your environment. So the first, as mentioned, from the vagina or via the C-section, where you would then, if you're born by through C-section, it would then be the skin microbiome. That's your first exposure. That's when you first start recruiting your microbes. But then after the birth, it's what you touch, eat, drink, suck on. You know, babies, they have a tendency to put everything in their mouth. Exactly. Yeah. So that's also the way that they recruit their microbes. But they also just do it by what they touch and not necessarily inject, because we also have microbes living on our skin, in our armpits, eyes, whatever.
So that's one of the things people have been talking about with the Antigel during the pandemic, that it's killing off bacteria from the hands, and that somehow that's bad. And that those would actually be counted into the microbiome.
Exactly. Because what we have of microbes on our skin is like an extra layer of protection. Your skin is already protecting you from pathogens and other harmful stuff. But the microbes do that too. And so if they're removed, you could have that, you know, less layer of protection.
Okay, so thank you microbes and thank you skin for protecting me. So we have this recruitment stage in the first couple of years. We run around, we suck on rocks, we eat sand, we do things, we get that exposure. Then, you know, what I think the first thing that comes to mind when I'd suggest when laymen hear there's a lot of bacteria, then they think, yuck, or that sounds dangerous, or that's something to prevent. So can you maybe walk us through some of that? What's the story here? Like when did we learn about bacteria not being all bad?
I think it's recently, actually, because for centuries we've been studying microbes, but we've seen them as the bad guys, the ones giving us diseases. And then I would say 30, 20 years ago it started changing. But I might be wrong. I don't necessarily remember if it is, you know, 30 years ago or 20. But it's recently. And I think it's very funny you mentioning that with the good and the bad, because in Denmark we have the shark tank as well. It's called Løvens Hule.
Yeah, the lion's den.
Exactly. And I recently saw the episode, I think it's the one from last week, where someone was in there trying to sell their product that would contain microbes, and it's a cleaning detergent containing microbes. That was their product. And they didn't really buy it, and they were confused about like the good and the bad. Sure, they're there, but is there anyone in between, and so on. And we definitely have a lot of different microbes, good, bad, and in between. We also, for example, have opportunistic pathogens. So those are pathogens that are not necessarily pathogenic, give you illnesses, but they can do it if the right condition is there.
So a pathogen is a bacteria that can make me ill.
Yes, but it could also be another microbe. Exactly.
Okay.
So it's the term we use to talk about which microbe that gives diseases.
Okay. So I feel like we have some sort of intro here where there's a lot of things to be confused about. That's okay. Let's keep it that way. Now you've explained to me as if you were explaining to an 11-year-old, and I feel like I was the brainiest 11-year-old in the room.
You were. You get A+.
Thank you. Can I go to high school then? So next up on our agenda is
well, do you mind? I was just quickly thinking of something I forgot, because when we develop our microbiome, it's important for me to also add that if you're breastfed, for example, that has a huge impact to whether or not you should develop autoimmune diseases, besides if you were born through C-section. And so there are many elements in this stage from when you're 1 to 3 that can, you know, affect your life later on. Because once we become adults, our microbiome is stabilized, usually. It's kind of settled. You can change it, but it will take a lot of effort. And then when we get older, to go to the end of the microbiome, it again gets less diversified. So the members, the microbes you have on you, they tend to die, so you don't have the same diversity. And you are, again, as an elder person, then more exposed to getting sick and to pathogens and so on. So I just forgot that maybe I didn't fully answer your question about the start. Yeah, about the start and the end and the different elements involved. So okay, I just wanted to add that in.
Now we have sort of the full life cycle. So you get born and then first couple of years, that's when you recruit the microbiome, then it's more or less set with deviations from that as you go through life, but it's basically your microbiome, the colony is basically built.
Exactly.
And then hitting some point in life, it starts kind of degenerating and becoming more homogenic, and that's basically when you get the exposure and a reduced sort of immune response, sort of in the later stages of life.
Exactly. And now you're ready for high school.
Now, okay, I'll enter high school then, and then I'm going to ask you why is the microbiome interesting and relevant for society to look at? I mean, I know you're getting a lot of funding for the project and it's sponsored externally. So why is this relevant?
Well, if I may first ask or answer the funding part, it's not me getting the funding.
It's your project.
Exactly. I'm a lucky person who applied for the PhD and got it. But it's funded by the Danish National Research Foundation, Danmarks Grundforskningsfond. And the center is the Center for Evolutionary Hologenomics. That's where I'm at doing my PhD. And what we kind of look into is how the microbes influence life and also how the microbes may have shaped evolution of life. And it can be any life. So we look at many different organisms and animals, including humans.
The fund that's financing this research, their job is to sort of make research about the complexity of the entire world, you could say, and then identify knowledge gaps, I suppose, and sort of have smart people help fill out those gaps. And that's what you're doing now.
Yeah.
So this is a relatively young science, you could say, or very young field that you're in. It hasn't been around for that long. So when we're looking at this, I remember you saying something about just how diverse the microbiome is. So if we looked at your microbiome and my microbiome, how many entities would there be in each colony, and how many would approximately be the same?
Approximately, you and I and our gut, for example, we only share, I think it's 10% the same.
Wow.
But I mean, when you think of how many genes you and I share in our genome, so the DNA we have, then it's still quite a lot like sharing in common with the microbiomes, the 10%. But you can also view it in the opposite way, saying, that's not a lot, like how come we only share 10%? But we as evolutionary biologists really see that as interesting because you and I share DNA at 99.99%.
Yeah, it's
a lot. Like we're almost identical, although we look so different, right? But we have gut microbes and other microbes living on us that are so different that may also play a part to why we look differently and why we act differently, why we eat differently. So they really are playing a huge role in this, I believe. But it is, as you said, a very new field. It emerged more or less 20, 30 years ago, thanks to next generation sequencing.
Let me just get this straight. You're saying that the bacteria that is in our gut is also sort of helping determine our appearance, not only genetics.
Well, it's still very, very new, the results. And I don't even think that you could necessarily say that they define, for example, your eye color or your tone of skin. I don't think it's necessarily that phenotype that they define. But we as biologists, we see phenotype as also the way you behave and how quickly, for example, you age and stuff like that. And scientists do believe that microbes influence that. For example, studies have shown that there is a correlation between which gut microbes you have and getting Alzheimer's or getting Parkinson's or getting other diseases that degenerate you and makes you older. So the bacteria in my gut are the ones to blame for my gray hair. Not my dad. I wish you could simplify that easily and I could say which microbes so you can just get rid of them.
I'll write them a letter.
do it.
We'll take it like that.
Or like chat to me in 10, 20 years from now. Maybe we have the answer.
See if there's something to...
Yes.
Okay. If we're only sharing just around 10% on average, one human to human, I'm assuming it works like that. How can we, I mean, are they not the ones that help us digest our food and sort of also prevent illnesses you mentioned and sort of be part of the immune system? How can we then have sort of 3 macronutrients that we're looking at in general sort of nutritional sciences? Do you expect that to change radically, and how much can we learn about that? I also understand there's a couple of thousand different types in each microbiome, so it sounds incredibly complex.
Yes, it is. But I do believe that it could move the field from where it is today. And we have, I mean, I was always going to sound like Trump, but billions and billions and billions.
Yes.
We have billions of microbes living in and on us. And therefore, I recently read that we actually have, I think it was 100 trillion microbial genes that can affect us. That's insane.
That's a Mickey Mouse number. I mean,
I know,
doesn't even...
But to try to compare it, we humans, we genetically in our DNA have 25,000 genes that we know codes for something. Mm-hmm. For example, your eye color or your skin. Whereas the microbes I just explained that live in and on us, they have 100 trillion genes. So it's 25,000 versus 100 trillions. So they really do have a lot to say, but what we're still trying to figure out is, does the microbes living in our gut that, for example, produces, let's say, serotonin, which is a neurotransmitter that can affect how you sleep and if you're feeling depressed.
Yeah, and it's, for laymen, it's the happy hormone, right?
Exactly.
It's what makes you happy.
Exactly. Many would be prescribed it if they, for example, are depressed. So it's exactly that. So we know that microbes in the gut, they produce serotonin. However, we also know that some microbes living in the soil produce serotonin. So it doesn't necessarily mean that those that are in our gut are doing it for our sake and are doing it because they consciously want to affect our behavior. And what's also important to keep in mind is that although they produce serotonin, that molecule itself can't directly go from our gut to our brain because
it's not accessible.
Yes, because of a specific barrier called the blood-brain barrier. However, microbes, they are also capable of producing the small, stone that gets built into serotonin. So they can produce an amino acid called tryptophan, which is built into serotonin. And that amino acid, tryptophan, it can go from the gut to the brain through the blood-brain barrier. So it's quite complicated, and that's why, for example, when I was talking to Alexander in the dyriske time, I didn't even go to this level of detail because it might throw someone off. And I think we should just maybe leave it there unless you feel later on going into it.
Maybe if I feel like accumulating my own intelligence again, then
exactly.
We can go there.
But it's definitely just important to keep in mind to understand the level of complexity and to understand how they can influence, but also how they cannot, because it's really at its infancy microbiome research.
We've been talking a lot about calories in is calories in sort of dietary restrictions and recommendations, and it seems that the caloric burn value is one of the very few things that nutritional science can actually agree exists and has an influence. Mm-hmm. So you'll need the number of calories in order to survive, and you'll need to reduce the amount of calories if you want to lose weight, for instance. Mm-hmm. And sort of for societal health, obesity is of course a major issue that needs addressing. But when we're looking at sort of dietary advice, dietary plans, I mean, is it down to the microbiome how we digest certain foods, or is it something else?
They definitely play a huge part in that. So we know that microbes, for example, specifically are also correlated with obesity. But it's still something that is very much debated. because some results don't necessarily overlap with others. However, I do believe that the microbes, they help us digest our food. It's not just something actually I believe, it's something we all believe and know of at the moment. But specifically, who is digesting what and at which level, and that is still not defined as far as I know. However,
but it makes sense because they're doing they're going through a respiratory process, right? So they have to eat something.
Yeah, they eat what you eat.
Yeah,
and
they eat my food.
Yes.
And break it down into smaller parts, and then it becomes accessible.
Yeah.
Through the intestines, etc. Right?
I mean, for example, they produce the B12 vitamin, and they also help break down fibers and so on. So they definitely do help.
Yeah. Maybe I'd like to take a few steps back actually, because now we've sort of dived quite deep in, and our listeners actually have no idea who you are. So maybe it would be good to sort of share your journey into studying the microbiome, and you can go as far back as when your own microbiome started and you were born, or whichever point you think is meaningful.
Yeah, I actually think it's very much related to what we just talked about, right? Because my interest in biology occurred quite late. I was in high school and I didn't know what I wanted to do, and so I took a gap year, tried to figure out what do I want. And I just gambled on biology, started on it, and I really got sucked in, but later in my bachelor. First it was just about partying and having fun.
It was the gin song.
But then I started having courses in microbiology and in cell biology. And I was like, damn, I can't believe that something so small, like a single cell, can have such a complicated life and is able to do so many different things. Like, we even talk about microbes having behavior, like themselves. Wow. So it's really, really complicated, and that really was enchanting and fascinating to me. And so I started nerding in a lot, and I got to meet Tom Gilbert, who's now my main supervisor. And we started collaborating, and I then later on stuck around and started doing my PhD. And my PhD is specifically about how the gut microbes, they can affect behavior in animals.
Yeah. And that's kind of the thing I wanted for you to sort of fold out. So you told me about this, how the microbiome is actually dictating behavior, and that's what you're sort of trying to investigate or prove or falsify. I don't know how you put that. And as I understand it, there's a potential that you can make a fecal transplant from one animal to another, and that can make them change from being subordinate to dominant, or aggressive to passive, or sort of not aggressive. I don't know what the antithesis is scientifically speaking, but
no, you're right.
It just sounds very science fiction-like, that, and also like something out of a comedy or something. You know, I can just see Seth Rogen being like, "Okay, so you did a shit transplant," and I mean, it's. Can you fold this out? Like, what's going on?
So specifically, when you mentioned dominance and aggression, it's because we have had a conversation per phone to discuss what are we going to talk about today, and it's because I work on two animals. I work with mice and dominance in mice, and I work with foxes and aggression in foxes. So that was why you gave those examples, just to clarify it. But it could be so many different things. We're specifically looking at these two behaviors because they have not yet been covered in mice or foxes. It's really a broad and wide field, and there are so many different behaviors that can be looked into, and I think that the interest to me was like, well, how the hell is this possible? Because it does sound so out there, like a science movie. But it is also now at a stage where we're looking mostly at correlations and trying to dive into causation. So seeing if there is like relationships that are not just we think this and that, but we can actually show that this does do that. So that's the stage where we're at now. And I am specifically mostly looking at the correlation part, and maybe it can later build into the causations. Yeah.
Okay, so it's the way I'm imagining this, and I don't remember if I'm making this up in my head or you explained it to me, but you take 2 mice and you have you figure out what their behavior is by letting them basically walk to the printer at the same speed, and then they go this way, that way, and one of them is going to sort of bow down and let the other one come through and then go into reverse and back out.
Yeah.
Right.
So specifically the mice, we test by having a 30 centimeter long transparent tube, and we take 2 mice at each end of the tube, release them at the same time. They meet at the middle of the tube, and then they fight, so to speak. Mice fight very gently, so it's more like a puff with the nose or whatever. And then the one who's the most dominant is able to push out the other mouse, and it would be considered the winner, the most dominant, and the one who was pushed out of the tube is considered the submissive one. So that's how we define it with dominance. However, with aggression, with the foxes, it was already predefined because they have been genetically bred in a way to be aggressive towards humans and also to be tame towards humans. So these are the 2 groups I'm looking at in foxes.
So they were being bred for certain purposes.
So they were already predefined in their behavior, so I didn't have to do an experimental test.
Yeah. You kind of accepted the breeding program as being the standard. Okay.
Yeah, it is. And then the fecal microbiota transplantations you were referring to is something we use when wanting to test out these causal relationships. So what we did was also to have feces from the dominant mice and feces from the submissive mice, and then we tried to swap it. So we gave the feces that was dominant to the submissive ones and the feces of the submissive mice to the dominant ones.
And here, I mean, I'm sorry, I can't help myself but asking, like, how percentage-wise, how much of their shit are you transplanting in this? Because I just can't get it to add up. If it's just like a small, doesn't it have to be all of it for it to be effective? Or how does that work?
Not necessarily, but it's still very debated. And I mean, there's so much going on with this procedure that the way I've done it is just one way to do it. But we took just a fraction of the poop and then we blended it and then we gave it to the mice. So it wasn't all of the poop collected, but a fraction of it.
Okay, and then, I mean,
also
just sort of, this also feels like a Donald Duck kind of thing where it's like there's someone who's pinching out and popping in, and like, is that actually a surgical procedure or how do you go about it?
It is. And also the method we used is of course from literature already out there. And
you didn't make it up on the spot.
No, exactly. Okay. It wasn't that freaky. But it is something that's done. I even know of doctors in Denmark who do it, like it's still in a trial phase as far as I believe. But where they would take, a donor who's healthy, who has a good, like healthy defined gut microbiome, take that feces, do the exact same, blend it, prepare it, take a fraction of it, or maybe even all of it, depending on which method. And then you would give it to the patient either orally, where they would have to take pills of poop, yes, or they would
eat it,
or they would get it via their anus. Yeah. So there are those 2 typical procedures.
Okay. I mean, it sounds super freaky, of course, but also fascinating that that's something we can do. If we're looking at sort of the microbiome versus behavior, what is it we can see? So what do we know today?
So, as mentioned previously, it's mostly correlations. But what we do see is, for example, that the gut microbiome influences depression, stress, autism. It could also be depression, Parkinson, Alzheimer's, like you name it. Yeah. It's a lot.
Okay.
And what
a lot of the moneymaker diseases, actually.
Yeah. And now we're looking into, as I mentioned before, like the causal relationships between the gut microbiome and a certain behavior. Yeah. And recently there was a super cool study published last year, actually, that showed that we actually can have microbiota-based therapeutics. They showed that autism, which can lead to either you having a hyperactive behavior or you having social deficits. They showed that mice, it was not in humans, but mice who were then autistic, once they were given a specific bacterial strain or a specific metabolite induced by microbes, then either or the bacteria or the metabolite, they would then, after getting it, act normal. So you had this mouse that was autistic, you give it the bacteria, no longer autistic. You give it the metabolite induced from bacteria, no longer autistic.
Wow.
Yeah. So, and this was a very, very scrutinized, well done study. Of course, it's just the one, so it's still something we need to verify, but it's really promising, and I definitely do believe we'll see in the next 10 to 20 years a huge shift of the way we do health treatments now and how we will do it in the future.
Amazing. And I think you could give me no better cliffhanger for because I would like to invite you for another episode.
Okay.
So I think let's wrap it up from here, and I'll just briefly summarize for our listeners, and say that so basically when we're born with the beginning of microbiome. It's a colony of all kinds of microorganisms that we recruit through the first 3 years of life. They basically start at the first barrier of our bodies, at our skin, and go all the way through mouth and all the way to the anus. And they protect us, but they also help dictate our behavior by making certain proteins available, for instance, for production of serotonin. They could be dictating how our genes are actually shaping the way we turn out as humans to a higher degree than our genetic material does it, or contributing. Maybe I'm slaughtering this a little bit.
Yeah. But
I slaughtered it, okay.
No, I just think, I mean, we as scientists are a bit conservative, right? And I think the way you explained it was great, and I really do hope that that's something we could maybe prove like fully in the future, but we're not quite there yet.
We're not certain.
No.
And that kind of calls for the two super interesting stages where we can see that there's reason to believe that it's also influencing behavior. That's under investigation. And last but not least, the potential for developing new therapeutics and new treatments, especially for anything mental, is quite vast, as recent publications have shown that a bacterial strain can actually put in parentheses or quotation marks, cure autism in mice. Yeah. Amazing. I look very much forward to diving into that. Thank you so much for being here, and thank you all for listening. We will continue producing these episodes, so do share, comment, like it, and tell us what you think. We're quite open to feedback and to taking on new guests or new themes. Of course, we're mostly touching upon how we can decarbonize healthcare.
And thank you as well for inviting me. It's been a pleasure.
You're more than welcome, anytime.